Hello.
Morning.
Sorry, I cannot listen your voice. Sorry. お世話になってます。はい。はい、すいません。はい。はい。はい。はい。はい。はい。はい。はい、大丈夫です。はい。はい。はい。はい。
Hello, can you hear me, sir?
はい。
Check.
そうですね。もう、おそらく過去にやったものがあると思うので、それはいただけると思っています。はい。
Hello.
はい。はい。
Check.
はい。はい。
Hello.
はい。わかりました。はい。はい。はい。はい。はい。はい。はい。
Hello.
はい。
Check.
はい。わかりました。はい。いえいえいえ。はい。初めてです。はい。
Check sound.
はい。はい。わかりました。はい。
Hello. Check.
そうですね。今のところ、ただ、ちょっとですね、まだ12月にどうも増えるっていう情報を聞いているので、学生からも。うちの研究室にもちょっと一人受けるっていう子がいるので、ちょっと増えるかなと思っています。はい。はい。はい。はい。はい。はい。わかりました。ありがとうございます。はい。ありがとうございます。よろしくお願いします。失礼します。よいしょ。ふうふう。 Test.
Hello, sir.
Hello. Yeah, okay. I can hear your voice clearly.
Okay. My name is Indrawan Permana, sir. I am a Vice Director of Graduate School, Palangkaraya University.
Yeah. My name is Mitsuru Hattori, Associate Professor at Nagasaki University. Yeah.
Okay, Professor Hattori. Konnichiwa.
Konnichiwa. Selamat siang. Kenalkan, nama saya
Yeah,
Mitsuru
yeah.
Hattori.
Oh yeah, nama saya Indrawan. You can speak Indonesia. So
Yeah,
I was in Toyohashi. I was in Toyohashi for 4 years studying my doctor.
Microbiology?
Toyohashi
University. Yeah. Technical University. Yeah. Toyohashi 技術大学. Yeah.
Yes,
I know. Yeah.
Yeah. And I'm so happy, sir, to have you here.
Yeah. Yeah, I'm happy too. Yeah.
So yeah, also with particularly in graduate school of Palangkaraya University.
Yeah. Sorry, sometimes internet network sometimes breaks your voice.
Yes.
Yeah.
Yes, I saw. I saw also here that the internet connection is not unstable. But hopefully we can finish this international lecture without any interruption.
Okay. Yeah.
We will start sharply at. We will start sharply at 9 o'clock. But the director of graduate school of Palangkaraya University and also Aswin Sensei will convey a short speech. So we will wait for maybe 15 minutes. After that, your turn. You will be the first, and then the third. And I hope that we can finish this international lecture by 12 o'clock.
Sorry, I cannot hear. The internet network is unstable. Please use chat.
Okay. I will chat.
Yeah. めっちゃ大丈夫かこれ。起きれた。
Yeah, I just check,
Yeah.
Professor Hattori. I just checked the connection.
Yeah.
In our site, it's also up and down.
Okay.
But now I can. Okay, I can see. I can see your screen, your slide.
Yeah. Can you see my lecture PowerPoint slide?
Yes,
Okay.
yes, I can.
Yeah.
I can see it
Okay.
now. That's perfect.
Okay. Thank you.
And Professor Hattori, if you don't mind, we need your slide presentation
Yeah.
to be sent to us. So we will give to all participants. Yeah.
Okay. The PDF or yeah. You prefer to the PDF or PowerPoint? Do you like
PowerPoint, sir.
PowerPoint? Okay.
PowerPoint is okay.
Okay.
No problem.
Okay. The file.
Oh, oleh Pak Indrawan keluar tadi akhirnya terpindah ke Pak Hattori.
Dalam bicara.
Okay, can you get the PowerPoint?
You sent to my email, right?
Yeah. No, the Zoom chat function.
Oh, Zoom chat. Okay. I will check it. Okay, now we see. Thank you very much, Professor Hattori.
Yeah, welcome.
Excuse me, Professor Hattori. We make a mistake. We should make you as a co-host instead of a host.
Overhost?
Yeah. Yes, we should make you as a co-host instead of host. So now you are host now.
Yes.
So
Okay.
if you don't mind, please give it back to us as
Yeah.
a host, and you will be a co-host. So we can regulate here.
Yeah. And okay. I just get out the okay. I finished the
That is our
meeting.
mistake. I'm sorry.
Yeah. Okay. Yeah.
Yeah. Okay. I am host now. Thank you very much. Thank you very much, Professor Hattori. I apologize for that. Kenapa ini? Kenapa? Oke.
Ini keberhenti Umar nih kan ada. Satu ini siapa yang mau?
Oh, jadi anu Bu, jadi siap lah.
Oh,
Jadi dengan Febri jadi. Kita akan mulai jadi itu Bu. Event tuh. Hello everyone, I would like to greet you. Saya ingin mengucapkan selamat datang kepada seluruh partisipan yang berasal dari Graduate School of Palangkaraya University, program pasca sarjana. Selamat datang dan silakan menunggu karena kita akan memulai acara ini. Sebentar lagi. Professor Hattori, if you are there, I would like to ask you, is Professor Wada already there? Wada tak? Rame lah Wada gitu. Hello, Professor Hattori and Professor Wada. If you can hear me. Halo. Halo. Apakah suara saya bisa didengar? Halo, selamat pagi. Oh. Halo. Halo.
Ya, jelas suaranya.
Jelas ya suaranya, Pak?
Jelas sekali, Pak.
Oke, baik. Professor Hattori, if you dare, you can hear my voice. And also, Professor Wada, can you hear me? Nggak usah gitu. Halo Pak Aswin. Ya. Professor Hattori, if you there.
Halo. Pandangan
Ya,
dengar saya.
Profesor Aswin, Dr. Aswin. Ya, saya bisa dengar, Pak.
Okay. I'm in Pulang Pisau now. On the way.
Okay. Okay. Professor Hattori is already in, but disconnected for a while, so I can hear him now. Dr. Aswin.
Yeah.
Hello.
I can hear you.
Okay. We will start the international lecture, and Director of Graduate School of Papaya University will give speech as an opening speech, and then Dr. Aswin as a head of LPPM will give a welcoming speech. Not too long, but make it as short as possible. Then Professor Hattori will start the lecture. Hello. Good morning, Professor Takeyuki Wada.
Hi, good morning. Nice to meet you.
Nice to meet you, sir.
Can you hear me?
Very clear, very clear.
Yes, I can hear you.
Yeah. Nice to meet you. Professor Hattori will come soon.
Thank you very much for coming.
Yeah, I can join.
Hattori-san iru?
Yeah.
Yeah.
Oh, Professor Hattori.
Okay.
Oh, you still there?
Yeah.
Okay. All speaker is already in, and we will start the international lecture, and the moderator will lead us to the opening session. Please.
Nah, kuman gak ketawa nih. Jelas. Kita nih. Oh.
Hello. Good morning. Morning, everyone. How are you?
Good morning. Fine.
Good morning.
Okay.
Okay.
Before we start the event, I will read some rules about this activity. Online International Public Lectures Mechanism. The online international public lecture is divided into 4 sessions. The first session is the opening session, presentation of the rules of the online public lecture and the opening ceremony. Second session is presentation from the speakers.
Zoom meeting international.
And the third session is a question and answer. But here there are several rules for asking. First, questions are written in the chat room. Please write questions during the presentations. Focus, non-wordy, non-repetitive questions that have been asked. Don't forget to write down the identity of the questioner, such as a name, study program, institution, and to whom the question is addressed. The host will display the selected question from the chat room in the question and answer sessions. The moderator will invite speaker to respond the question. And next, the online attendance links will be shared after the third speakers. The final session is closings. And before that, we will get a photo together. And next is rules of online public lectures. Please ask questions according to the theme and focus on the problems. No interruption from participant, and please turn off the speakers during the presentation so as not to be storming. Please don't leave Zoom during the activity so that the online international public lectures run conductive and respect the science councils. Thus, ladies and gentlemen, to be understood for the running well of our public lectures today, thank you very much. But there is special notes for participants here. The full name on the attendance form will be used in the e-certificate. Please make sure the name submitted on the registration form is correct. The email address will be used for a certificate delivery purposes. Please ensure that the email sent via this registration form is active. Participants who get a certificate via email who are registered at attendance form are participants who follow the event until the end and fill out the attendance form, which will be given toward the end of the event. Information on the Zoom meeting link and YouTube streaming will be delivered by the committee through the WIAG group of the online international public lectures. Participants are not allowed to share ID and password without permission, and participants are requested to join no later than 10 minutes before the activity start. Participants are requested not to use a virtual display screen that interferes during the activity and is not allowed to turn on the speaker while the speaker is presenting. Thank you. Healthy greetings. Virtual International Public Lecture on Ecology, Genetic Diversity, and Land Use, Thursday, October 22nd, 2020, held by postgraduate programs of Palangkaraya University and Nagasaki University, will be start in a few moments. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. UPR Jaya Raya, Harati Bacahaya Kilau Bintang. Thank you so much for being here. Let me begin by giving you a warm welcome to the International Public Lectures of Postgraduate Programs of Palangkaraya University and Nagasaki University. Previously, I want to greet the student from Postgraduate Program Palangkaraya University. How are you? I hope you are all well. Okay, for student from Nagasaki University, Nagasaki Daigaku no Gakusei no minasan, yokoso, o genki desu ka? Hai, o genki desu. Watashi mo ii desu. Watashi no namae wa Dr. Misurita desu. That's all I can, so don't ask me further in Japanese. Okay, I am your moderator today. It's such an honor to welcome you here, ladies and gentlemen. First, let's pray and praise to God because of His blessing and mercy. Then we can come together without any obstacle, with healthy condition in this international public lecture, which are held by postgraduate programs of Palangkaraya University from Indonesia and Nagasaki University from Japan. The Excellency Director of Postgraduate Programs of Palangkaraya University, Ibu Professor Dr. Insinyur Yatri Ludang MP, the Honorable, the First Vice Director, Ibu Dr. Evi Veronica Elbar, the Honorable, the Second Vice Director, Bapak Dr. Indrawan P. Kamis, as the speakers also, and the Excellency, the Head of LPPM of Palangkaraya University, Bapak Dr. Insinyur Aswin Usup, the Honorable all of Head and Secretaries of Study Programs in the milieu of Postgraduate Programs of Palangkaraya University, the distinguished speakers, Professor Mitsuru Hattori, PhD, and Professor Minoru Wada, PhD from Nagasaki University, Japan. It's such an honor to welcome you here. And last but not least, to the whole of lecturers and students of postgraduate programs of Palangkaraya University from Indonesia and students of Nagasaki University from Japan that I love and I'm proud of you. Now, on this special event, we have several agendas. As follows, the first session is opening, the second session is opening speech, and the third session is presentation time from all the speakers. The fourth is question and answer question, and the fourth session is closing. Well, ladies and gentlemen, let's begin our agenda by opening speech. We invite directors of postgraduate programs of Palangkaraya University to deliver the opening speech as well as open officially these international public lectures. Ibu Profesor Dr. Insinyur Yetri Ludang. Okay, wait a minute. There is a troubles up and down. Wait a moment, please. We invite the Director of Postgraduate Programs of Palangkaraya University to deliver the opening speech as well as open official these international public lectures. Ibu Profesor Dr. Insinyur Yetri Ludang MP, the floor is yours.
Thank you, moderator. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Thank you, moderator. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Good morning. Tabe salamat lingunalatai, salam sahajut ngarendem malempang, adil katalino, bacuramin kasaruga, basengat kajubata, arus, arus, arus. Ethnic greeting of Dayak Ngaju. The Honorable Vice Director for Academic Student Alumni Affair of Postgraduate Program of Palangkaraya University, Ibu Dr. Evi Veronica Elbar. The Honorable Vice Directors for General Affairs and Finance of Postgraduate Program of Palangkaraya University, Bapak Dr. Indrawan P. Kamis. The Honorable the Head of Research and Community Service Institution of Palangkaraya University, Bapak Dr. Aswin Usup. The Honorable all of Head and Secretaries of Study Programs of Postgraduate Program of Palangkaraya University. The distinguished speaker, Prof. Mitsuru Hattori, PhD, and Professor Minoru Wada, PhD, from Nagasaki University, Japan. Congratulations, nice to meet you virtually. And the last but not the least, to the whole of lecturer and student of postgraduate of Palangkaraya University, Indonesia, and the student of Nagasaki University from Japan that I love who join to this valuable event. Welcome to this international public lectures event virtually. I am Professor Dr. Insinyur Yetri Ludang MP, standing here as the Director of Postgraduate Program of Palangkaraya University. I am quite happy on the effort to the implementation of this prestigious seminar under the theme Ecology, Genetic Diversity, and Land Use, which is held by Postgraduate Program Palangkaraya University and Nagasaki University. My highly appreciation to the head and the member of the International Public Lecturer Committee who have made a great effort, then we can escalate this great international public lecture. As the director of postgraduate program of Palangkaraya University, I am really respect to this event and I hope this is as far excellent. And next on 2020, I wish we can hold the most prestigious international seminar event. Now, let me explain some point view about postgraduate program of Palangkaraya University. The postgraduate program is one of master and doctor at Palangkaraya University. It consists of one doctoral study program in environmental science and 11 master study programs. They are Master of Management, Natural Resources and Environmental Management, Master of Law, Master of Economic, English Education, of School Education, Chemistry Education, Biology Education, Basic Education, Social Education, and Economic Education. The lecturer consists of 22 professors and 93 doctors. The vision of the UPR postgraduate program is to produce best quality human resources based on local excellence. Local excellence mean here is related to peatland. Postgraduate program of UPR is always want to expand access and partnership and collaborate with postgraduate students at national and international level, one of which is with Nagasaki University as well as with the regional government, the business world, professional association, and other. The education standard in the postgraduate program follow the Indonesian Ministry of Education and Classes National Standard. Thus, the international public lectures on topic ecology, genetic diversity, and land use held by postgraduate program Palangkaraya University and Nagasaki University, I declare it is open. Thank you. Before I end my opening speech, let's together for a woman watch the video of Palangkaraya University Natural Pit Forest in line with the postgraduate program's visions of local excellence, namely Pit. Why? Sabar.
Program Pasca Sarjana Universitas Palangkaraya menghasilkan SDM yang berkualitas sungguh, berintegritas ilmiah sebagai ahli serta praktisi, kreatif, inovatif, transformatif, analisa tajam dan komprehensif. Majulah program pasca sarjana Universitas Palangkaraya.
Sudah. Yeah. Thank you. Finally, I hope this international public lecture are running well, keeping some contribution and beneficial value to the whole of audience who have joined in this event. I give it back to moderator.
Thank you for the director's insight in the opening speech. Ladies and gentlemen, next we invite the head of LPPM of Palangkaraya University to give a speech. Bapak Dr. Insinyur Aswin Usup. Time is yours, sirs.
Yeah, thank you, Miss Rita, for the time opportunity.
You're welcome.
I'm so sorry nowadays. I'm still on the way to Pulang Pisau, and then I stop in one warung here, and I can enjoy joining
That's
with you.
great. That's great.
Yeah. Yeah, I will going to Balanti, about 170 kilometers from Palangkaraya. Yeah. But thank you very much for opportunity today. Yeah, I would like to say thank you. Yeah, the Honorable Professor Dr. Yetri Rudang for nice speech. The Director of Postgraduate Program University of Palangkaraya, who has provided the opportunity and time for organizing this international public lecture to be held today. The Honorable Professor Mitsuru Hattori from Nagasaki University, who has conducted research on pest and disease, yeah, of food crop in Central Kalimantan, in collaboration with lecturer and also student of Palangkaraya University. And also, I would like to say thank you for the Honorable Professor Takayuki Wada from the Graduate School of Human Life Science, Osaka City University, from his valuable time to teach in our online lecture, to upgrade our master and doctor student, especially on basis molecular biology, genetics, and other. Greeting from Palangkaraya, and I hope to visit and do research in Central Kalimantan in next future. And also, Dr. Indrawan, this my good friend, Vice Director of Graduate School, who organizing this lecture and also provide the lecture on the land use in Central Kalimantan, especially PO point from planological concept. And I respect and proud are all of master and doctor student, both Palangkaraya University and also Nagasaki University. Honorable guests, ladies and gentlemen, today I am so happy can be joining with all of you to conduct this international lecture. This is maybe the first time with Nagasaki University under the collaboration between Faculty of Agriculture of Palangkaraya University and Graduate School of Environmental Science of Nagasaki University. This activity, I hope, can be valuable both from University of Palangkaraya and also Nagasaki University. In future, I strong hope can be established an international class between Indonesia and Japan, especially between Nagasaki and Palangkaraya University. If we can realize this, more hope can be improved level of Palangkaraya University to be a great. Yeah? This is our dream, hopefully to be reality. Honorable guests, ladies and gentlemen, in Central Kalimantan, we have abundant and huge area of peatland area, and most of this peatland has been degraded and firing every year. But University of Palangkaraya still have 50,000 hectares of dense tropical peatland forest for research and education. We call it natural laboratory under Sintrop and Sabangau National Park. Nowadays, in the other hand, in Central Kalimantan has been announced and starting producing the coastal peat for national food security, about 300,000 hectares developed within this 2020 fiscal year. For that, we really need the support from all of the parties, including Nagasaki University, and also from Osaka City University. Through Dr. Mitsuru Hattori and also Professor Wada, we need to prepare reliable human resources who graduate from Palangkaraya University to better understand the importance of sustainable environmental management. Honorable guests, ladies and gentlemen, thank you very much for holding of this international lecture. Hopefully, this is not the first. Hopefully, this not be to the first and the last time. I strong hope that this collaboration can continue to the university level, and some of our students can be continue study master and doctor in Nagasaki and also in Osaka City University. Warm greeting to the President Nagasaki University and all of the team who have conducted the research in Palangkaraya. Congratulations on joining this lecture. Minasang, kokoryoko. Arigato gozaimashita. Dare kaga soreo kini narakatara. Gomenasai. Sonani, arigato gozaimashita. Thank you very much.
Hai, arigato gozaimasu. Okay, thank you, Bapak Dr. Insinyur Aswin Usop. And ladies and gentlemen, before we come to the presentation sessions, our beloved directors will entertain us with her beautiful voice. She will sing a song from Japan's Kokoro no Tomo.
Yes.
Okay, please.
Yes. I would like to close this opening speech with a song of Japanese, Kokoro no Tomo. I apologize if my voice not so good to hear. Thank you.
Okay, thank you, Ibu Director, who has entertained us with her voice. And ladies and gentlemen, next we come to the main agenda. I convey you that our event today took the themes ecology, genetic diversity, and land use. These themes is very contextual to our current situations. It's because the damage of ecology in our surrounding every time is getting increased. So by implementing international public lecture, it is one of our awareness toward the importance of our ecology. And to know more about the issues, we need to invite the professional people based on their competence. Let's applause for all of guest lectures today. Thank you. Okay, it's important to inform you to all the participants first that in the question and answer sessions, we will collect the question from the chat room. So while listening to the speaker's presentations, you can write the question in the chat rooms. Don't forget to write your name and study programs and to whom question is addressed also. Ladies and gentlemen, the first guest lecturer who will deliver his paper under the title Introductions of Ecology is Professor Mitsuru Hattori, PhD, from Nagasaki University. The floor is yours, sir. Hello?
Oh,
Okay.
okay. I will start my lecture, but please permit sharing the
yes.
my
Yes,
slide.
you can share your slides by yourself or by our host.
Yeah.
By yourself.
But I cannot share
You
the
can.
slide. And please
make him co-host. Make
please
props.
host permit.
Okay. Okay, wait. Okay. You are already become a co-host now.
Okay.
You can share now.
Okay. Can you see my slide? Okay. Okay. Selamat siang, kenalkan, nama saya Mitsuru Hattori. My name is Mitsuru Hattori from Nagasaki University. I'm so glad to do today's lecture for you. I'm surprised as a large number of participants in this lecture. Thank you for listening to my lecture. I'm going to talk about introduction to ecology in this lecture. When you have any questions, don't hesitate. Ask your question to me using the chat function of Zoom. Before the main team, let me introduce myself. My research team is ecology and evolution of species interactions. I will talk about species interactions soon. I have studied the adaptation of organisms through species interaction since undergraduate. Especially, I focus on prey and predator interaction and pollination mutualism. Both interactions are important for the survival of organisms. So, first theme is what is ecology? We use term ecology easily, but do we know definition of ecology? Maybe no. Here, I'm going to talk about definition of ecology. On the planet, many organisms live. Each organism shows a specific distribution. For example, orangutan lives only in around Indonesia. On the other hand, polar bear lives only in around Arctic. Why does not polar bear live in Indonesia? Ecology is a study for seeking this answer. So the definition of ecology is a study for understanding the distribution and abundance of organisms. Distribution and abundance of organisms are influenced by various environmental factors. We can roughly divide environmental factors into two types as abiotic factors and biotic factors. Abiotic factors are non-living chemical and physical factors of the environment, such as sunlight, rainfall, temperature. Biotic factors are any living components. These factors are equal to biological interactions. Biological interactions contain interaction within species and interaction between species. Interaction within species is communication between individuals of same species. The interaction contains competition for food, space, mating partner, and any limited resources. On the other hand, interaction between species is communication between different species. This lecture is going to focus on interaction between species because my interest is it. The outcome in biological interactions can be classified based on positive and negative. The positive means that species get some benefit from other species through biological interactions. The benefit represents getting energy, increasing number of children, and saving their life. The negative means that species lose their some resources by other species through biological interactions. If two species give positive impact to each other, this relationship is called mutualism. This picture is an example of mutualism. Relationship between bee and flower is the most famous relationship of mutualism. Here, let me explain the good examples of mutualism. A first example is mutualism in ant and aphid. Aphid are white insects shown in the right photo. They have a lot of enemies which eat them because basically they do not have good defensive ability. From this reason, many of them should depend on the other organisms for saving their life. Interestingly, aphids have an ability of emitting sugar from their body. This ability is actually useful for their protection with ants. Ants always look for sugar because they need a large amount of sugar for their activity. Once they find source of sugar, they guard it from other animals. So aphids use their ability and ants' character for their protection. Then ants are also happy to get sugar from aphids. Overall, in aphid and ant mutualism, aphids employ their bodyguard paying their sugar, while ants cost their working power for getting sugar. Next, let me talk about another example of mutualism. Pollination is a transport of pollen between plants by bee. To make their seed, they need to send their pollen to other individuals. So plants need bees to bring their pollen into other partners in reproductive process. But bees do not bring pollen without any reward. Plants have an ability of emitting nectar on their flowers. They use this ability for pollination. bees want sugar for their activity. When the bees visit the flowers for nectar, the bees take their pollen without awareness, and then the pollen can be carried to other plants by the bees. Overall, mutualism in pollination, flowers employ their transporter paying their nectar, while bees cost their working power for getting nectar. So let me introduce example of impact of mutualism on abundance of organisms. Pollination helps plant to make the next generation. So if pollinator decreases, many plants decrease their population. So next, I focus on prey and predator interaction. Prey and predator interaction is the most common in nature world because almost animals must eat other organisms for their lives. In this table, species 1 is called prey. Prey is eaten by other species. Species 1 receives negative effect from species 2. To avoid these negative effects, preys have various defensive characters. On the other hand, species 2 is called predator. Predator eats other organisms for getting their energy. Species 2 gets positive effects from species 1. To increase their positive effect, predators also show various characters. So prey and predator interaction is a survival wave between two species. Here, I'm going to explain about impact of prey and predator interaction on abundance of organisms. Interaction between rabbits and lynx is a good example for understanding the impact of prey and predator interaction on abundance organisms. Snowshoe hare is common rabbit in the US. This rabbit is prey of lynx. Lynx is a specialist predator of rabbits. This graph shows annual changes in the number of rabbits and lynx. Green line presents the change in the number of rabbit individuals. On the other hand, red line presents the change in the number of lynx individuals. Their abundances shows tightly linked to each other. But both number of individuals are misaligned. Their number of individuals shows mismatch. Please see this point. The top of green line, after the top of green line, the red line top of comes. Both number of individuals are mismatched. This gap caused from their prey and predator interaction. The lynx numbers follow the rabbit numbers, crash after a lag of 1 to 2 years. As rabbit numbers start to decline, lynx continue to eat well because they can easily catch a rabbit. So their abundance shows cyclic pattern. Rabbits eat lynx. Rabbit decrease their number of individuals. After the decreasing rabbits, lynx of number of individuals decrease. In the next part, I'm going to discuss with students how to use the knowledge of ecology for our lives. I prepared a theme for discussion. The theme is what is useful biological interaction for our lives. please give your opinion after thinking time a few minutes, and give me your opinion by using chat function of Zoom. So please give me, and don't hesitate, and give me your opinion about what is useful biological interaction for our lives.
Okay, participants, there is a question from Mr. Mitsuru Hattori. There is in the chat rooms. Ada pertanyaan dari Mr. Hattori di chat room. Mungkin ada yang ingin memberikan respon. Please give me opinion about what is useful biological interaction for our lives. Mungkin ada yang ingin memberikan tanggapan dari partisipan. Apa kegunaan dari interaksi biologi untuk kehidupan kita? Anybody? Ada yang ingin memberikan respon? Mungkin yang dari biologi.
Okay, my student answered the question. She said that prey-predator interaction between pest insects and their predator. Oh, and the other answer is he answered that it will bring a mutual benefit, for example, between bee, butterfly, and flower. Yeah, okay, very good answer. So, prey and predator interaction is useful for our lives. Predator eat pest insects. This photo shows ladybird eats pest insect of sugarcane. Predator interactions can prevent pest damage and increase crop production. Japanese farmers prevent pest damage by using natural predator interactions without using chemical pesticide. Okay, yeah, and other answer is very good. So mutualism is useful too for our living. Most plants, such as tomato and banana, must be helped by must be helped by pollinator animals for making their fruits. Mutualism between plants and pollinators can increase crop production. Yeah, then yeah. So let me summarize this part. In this part, I focus on biological interactions in the nature. The biological interactions help our activities such as agriculture. In the next part, I'm going to talk about examples of the application of ecology for agriculture. So next theme is applying ecological knowledge. Here, I'm going to talk about the application of biological interactions for our lives. Ecosystems give us various benefits. For example, we can get woods, food, and water from forest ecosystems for our lives. This benefit is called ecosystem service. This term means every benefit that human gets from ecosystems. This term is promoted by Millennium Ecosystem Assessment. The service were classified into 4 categories: provisioning, cultural, regulating, and supporting service. Here, let me introduce the ecosystem service shortly. First service are provision service. Provisioning service are defined as a supply of goods that directly benefit people, such as food, timber, and fuel, and fresh water, and so on. Next is cultural service. Cultural service are defined as the non-material benefits that obtain from ecosystem through spiritual arrangement. For example, knowledge systems, social relations, and realistic value. Next is regulating service. Regulating service are defined as the benefits obtained from the regulation of ecosystem processes, including the including air quality maintenance, climate regulation. Finally, supporting services are defined as the production of other ecosystem services. So I think that the most important ecosystem service is regulating service. Especially pest control has a key role for agriculture in the future. Here, let me talk about an example of biological control for agriculture in Japan. Biological control is included regulation service in ecosystem service. This control is the regulation of the population density of pests by predators. For example, larvae and the adults of ladybird eat some pests on tomato and the eggplant in the wild. A Japanese company is applying ladybird to prevent pest damage. The Japanese company sells tent top for Japanese farmers. This tent top contains small larvae of ladybirds. When Japanese farmers find pests, they use tent top. This biological pesticide is very eco-friendly because ladybirds eat only pests and are not harmful to humans. On the other hand, chemical pesticides kill every animal, including predator of pests. Furthermore, chemical pesticides sometimes damages humans and pollute some ecosystems. I think that we have to use biological pesticides as much as possible to conserve environments. Next, I'm going to introduce to the role of forest on agriculture in Kenya coffee plantation. Kenya is one of the most coffee production countries. In Kenya, for coffee production, forests were changed to the plantation. However, some researchers related that the existence of forests helps to produce coffee through biological control by forestry birds and bats. Karps and his colleague tried to exclude birds and bats from coffee wood. This graph shows the pest control effect of birds and bats on pest damage of coffee. White birds shows pest damage in the natural condition, including birds and bats pest regulation service. Blue birds show pest damage in only bird excluded conditions. And we can see that pest damage in only bird excluded condition was bigger than the natural condition. On the other hand, pest damage in bat excluded condition was not bigger than the natural condition.
Ya, kamu mau, Mbak.
So birds can help to increase coffee production through eating pest insects. Furthermore, natural forest was important for pest control by birds because natural forest was habitat of birds. This graph shows the relationship between forest cover around plantation and bird abundance in coffee plantation. As you can see from this graph, the forest area correlated with the bird abundance in the coffee plantations. Thus, this graph shows that coffee plantation with large forest can prevent pest damage by birds. Therefore, decreasing natural forest means that agricultural production also decreases. If we recover agricultural production without pest control of the forest, we have to buy and use more chemical pesticides. This situation can lead to more poverty and more health deterioration of farmers. As I was saying, forest is important for improving agricultural productivity. In this situation, I believe that this story is true also in Indonesia. However, the forest cover area is decreasing by a forest fire and making plantation. And this picture shows a forest cover area in 2001 in Kandy Mountain. On the other hand, right picture shows a forest cover area in 2014. Forest cover area decreasing significantly recently. I predict that decreasing forest area cause pandemic of pest insects because predators may decrease along with deforestation. In fact, diversity of birds and insects in the plantation is 77% and 83% less than primary forest. primary forest and the oil palm plantation, forest was the diversity of forest was decreased significantly over the 70%. Thus, making plantation degrade various ecosystem service. For example, rice fields with a forest can prevent pest damage by forestry predator, but rice fields without forest may suffer from pest insect damage. In 2019, we observed community structure of insects and rice damage by pest insects in the Kapwas rice field. Table 1 shows our result. Hampangau and shrimps were most abundant. L. oratorius means Hampangau. Hampangau and shrimp species were most abundant. Surprisingly, we found Hampangau and shrimps mainly, but we found only few the brown plant hopper and the adults of stem borer. Furthermore, the damage by Hampangau was larger than 10% yield loss. These results suggest that big environmental change occurs in Kalimantan because Hampangau was not famous pest insect in Southeast Asia that has been reported. However, in Kalimantan, damage to rice by Hampangau is very serious. We have to make the method for preventing damage for Hampangau. So, next discussion theme, how do we take countermeasures for Hampangau? Please give me your idea. Give me your idea by using the chat function of Zoom. Please don't hesitate to answer your opinion. Please use chat function of Zoom. Okay, as my student answered, the pesticide or the using bath smell. So Hampangau can emit the bath smell. Yeah, this bath smell is maybe around pheromone. When Hampangau is attacked by its enemy, Hampangau emits bad smell. Hampangau's smell informs other individuals about danger. And this smell is called as around pheromone. When Hampangau feels a smell, Hampangao escapes to other place immediately. If this is correct, rice farmers can mitigate the damage from Hampangao by putting crushed Hampangao on the rice field. I believe that this method is very cheap and eco-friendly method for preventing damage of Hampangao. But we have to study the around pheromone of Hampangao and the effect of around pheromone on the rice field. Yeah, pesticide is very useful. Chemical pesticide is very useful, but the Hampangao can evolve the resistance of chemical pesticides immediately, because Hampangao have the ability of the evolution of resistance of chemical pesticide. Now, Hampangao can survive many under the spraying chemical pesticide. I will try to prevent by using the pheromone of Hampangao in Central Kalimantan. But under COVID-19 situation is difficult for me, and it is difficult. Yeah. So next year or after COVID situation, I try to prevent damage using the bad smell. So finally, let me summarize this lecture. Our lives need ecosystem service from nature. However, overusing natural resources is continuing all over the world. Therefore, we must stop to overuse and to destroy natural resources for a sustainable society. Thank you for listening. Thank you for listening to my lecture. I finished this lecture. Please, speaker Wada, Professor Wada Minor. And I will answer the question by using chat function. Yeah.
Prof. Mitsuru Hattori, are you finished with your presentations? Have you finished?
Yeah.
Yes, okay. Thank you. Prof. Mitsuru
You're welcome.
Hattori, PhD.
Yeah, thank you for
Give a
listening.
big applause for him. For the next guest speakers, ladies and gentlemen, please join me in welcoming Professor Minoru Wada, PhD, who will deliver his paper with the title, What is Genetic Diversity and Introduction of Molecular Evolutions and Genotyping. Time is yours.
Hello? Can you listen to me?
Yes.
Yeah, okay. Okay, I'm next speaker related to the genotyping and something genetic diversity. Sorry, my name is slightly different from the information. My name is Takayuki, and I just moved to another university from Nagasaki University last April. So I want to now I'm connecting from the Osaka University, Osaka City University to the Palanganaya University now. I think this is only the good point by the pandemic of COVID-19. And I like to visit the many places involved, especially in East Asian countries as a collaboration that unfortunately now we cannot. But fortunately, we have a method to connect directly, like now. Then it's my pleasure to share the information between us and although there are many long distance. It's my pleasure to have a lecture even such direction distance. Okay. Now I would like to have my introduction, self-introduction, because as I said, I just moved to the another university from the Nagasaki University. Actually, I was in Nagasaki University by last April. Then after that, I changed my position to the Osaka City University, but I still keep in touch with Professor Mitsu Hattori as a collaborator related to the ecology. In my research target is actually the public health and microbiology and infectious disease actually. Then although the Professor Hattori kindly have a relationship with me, related to some kind of the transmission of bacteria and within the ecosystem, and it's a very good challenging target. And so that he asked me to have some lecture in this opportunity, and then now I want to share the basic backgrounds of the genotyping and the genetic diversity. Then because I have some different kind of background with Professor Hattori. So now today's topic, I want to separate some kind of three topics, roughly. Then the first topic is background of the basic molecular genetics, or some kind of the molecular biology, which means what kind of molecular may be is related to our life activities, and after that we can understand what kind of some kind of the genetic diversity and the genotyping. This is a method to classify the lives based on the genetic diversity. So before I talk about such kind of the genetic information, I would like to focus on the basic situation of the molecular genetics or molecular biology. Anyway, this is very simple information about the molecular biology. Maybe almost all participants know about that. This is the very important background, so that I want to share. As we know, that we are life. Many kinds of lives. There are many kinds of lives species on Earth, and that's very complicated. Very complicated. This picture is a monkey in Japan. They can enjoy the hot spring in winter seasons. Then it's very complicated lives. But we know that the molecular is very simple in some classes of chemistry, like the hydrogen or the oxygen and the nitrogen, water, and so on. They're very simple. So that we need to consider what kind of the discrepancy, what kind of the background between the discrepancy between the simple molecular and the life systems. Now, basically, we can explain the complicated background of lives based on the central dogma. Central dogma include some molecules, very big molecules, the three important molecules, DNA and RNA and the protein. The protein is the most important molecule from the viewpoint of the effect of life activities. Because the proteins catalyze the many kind of the chemical reactions, including the get energies and make our children and the proliferation and so on. And of course, the cell activities also also based on the catalytic activities of proteins. Then, but such proteins should be designed by the blueprints, which means the DNA. DNA contains the information of what kind of sequence of peptide can become proteins. This is a summary of the information flow of information. The DNA, as you know, DNA includes the 4 nucleotides. This means adenine, guanine, cytosine, and thymine. Such 4 nucleotides has a sequence within our genes. Such sequence translate to the message. Translate to the protein sequence via messenger RNA. Then it's very important to understand such flow before considering the difference of DNA in our body. And the 4 nucleotide sequence can change to the three numbers of letters. Then the complex, the 64 triplets shows the information about each amino acid. After that, 20 amino acids, sequence of 20 amino acids can be designed by the DNA sequence. This is a central dogma. So the DNA is a blueprint of our life, and they are transcribed to the messenger RNA. After that, the information changed to the sequence of amino acids that become a protein. Then the DNA sequence is our background of the complicated information. And then it's very important to consider about the replication. DNA is easy to proliferate, the 1, 2, 2, 2, 2, 4, and so on. So that DNA can become a background of our proliferation activity. So that the DNA to protein is lateral. And then the DNA is very flow is very important to proliferate. So the DNA sequence comprise the genome sequence. Our human genome is about 30 gigabase pairs. Very big. And our all genes are written in the whole genome sequence. And of course, almost all life species have their own genome. So the bacterial genome, the size of bacterial genome about as like here, it's a very smaller than human genome, but they have actually their own genome. And of course, the viral genome also including, also existing. The virus also very small, but and then they need to use our cells to proliferate. But they have their own genome. Then they can become, they can proliferate their own descendants based on the sequence of DNA. Then that's a background. The size is very general information. Actually, there's some kind of exception, but the size of genome correspondent with the complexity of each life. So our genome is so huge, and although the bacterial genome is so small, and the viral genome is much smaller than the bacteria or some kind of the eukaryote system. So and also the genome include many kind of genes. Each gene has a function. Each function like some kind of unfortunately the 200 more than 200% the function is still unknown. That in other words, 75% or so of genes have already known their own functions within our body. Some information as like here, then so the very easy function is as like the nucleic acid binding, which means the interaction between the protein and the nucleotide, the DNA or messenger RNA or so. And the signaling molecules means that we can detect the difference of change of environment, and we can feel the hot or cool and so on. Such a signal should be translated from the skin to the nerve system, and if we touch fire, we need to escape. Then such signal should be controlled. The very complex process, but the protein can construct such an interaction. Finally, as a harmonization of the whole activity of enzyme. Then anyway, the gene have each function, and such the mixture or the total function comprise the life activity. Such genes are written in DNA. The DNA is very easy to proliferate. According to the time passing, that we make our descendants by the proliferation of DNA. The originally, on the Earth, the very simple life was there, maybe, probably. After that, they proliferate and evolve. After that, the current situation occurred. The human living, not only human, the many lives share on Earth. Not only the very complex animals, but also bacteria and virus and so on. Basically, the central dogma is based on then we can understand why DNA is important. Why the life activity is based on the DNA sequence and so on. And such DNA can proliferate on Earth with long time. Okay, this is a very important background. Under this background, we can talk about the genetic diversity. So genetic diversity, as we know, there are difference not only between species, but also intraspecies. We are human, and we are different. And such difference are driven by many kind of backgrounds. The genetic diversity is one of the most important difference to make such difference. This is a slight species difference between the species by species. Many monkey species different. They can make the descendants within species, and then each species have different DNA backgrounds. But within interspecies, there is difference, of course. In some case, some tomato have the large one, and some sweet or sour, and so on. Such intraspecific difference also based on the genetic changes. This figure means that this is a strain of the tuberculosis. Do you know tuberculosis? This is the airborne diseases. In some cases, it infect with our lung and show the symptom. My research target actually focus on the genetic diversity of tuberculosis in the last several decades. Still now, I continue to do research on this one. We cannot discriminate the species by species from their appearance, but they actually different, very different. But when we check the DNA sequence, so anyway, such a variation always occurs. Then why such variation generated is important. During the proliferation of DNA, the duplication of DNA, in some cases, the DNA copy should include the mismatching, miscopies, the duplication by duplication. So if even if the original sequence is existing, after duplication, each descendant may have the difference. If such difference causes the change of genes, the species become different. Such accumulation of mutation can cause the genetic diversity. Finally, even the one bacteria separate to the two bacteria, the mutation can occur. 2 generations, 3 generations, and finally, they have the very broad genetic diversity. Then after that, the gene may change. When the gene changes, the appearance or some kind of the phenotype, which means the biological change difference, can be found. This is some kind of the mutation, the color, and all the I don't remember the name of the species. Anyway, a peacock. And then, for example, this is an example of the mutations. The cat. In Nagasaki, there are many cats in town. And in Japan, most of the cats have long tails. But when I become an assistant professor in Nagasaki University, I was very excited to see the very teeny tail, very long, small tail. This had the mutation occurred in the population. And in some, this is one paper about the mutation of shape of tail in cat. This paper focus on the small small group of cats, the Manx cats. It's one of the brand of the England. They have the small tails, very very short tail. And then it's based on some kind of the mutation within one gene. Actually, historically, the Nagasaki was a very important port to have world trade in several hundred years ago. Then at that time, the ships moved to the European countries and then the Indian Ocean and to go to the Indonesia, Philippines, and finally Japan. Then at that time, the many the sailors put the cats on their ship and during the movement, and then many cats from Indonesia and the Philippines and Kalimantan Island to move to Nagasaki. Then I know that some many cats in Indonesia have the short tail cat. Maybe such relationship is existing within the relations cats. That's based on the mutation of the one genes and so on. But we cannot estimate the historical origins because now we live in 2020. That we can analyze only the current situation. This means, and so this is the picture about that. We can collect the many samples as like here. This is an example of the DNA sequence. Yes, this is a current situation, but probably they have common ancestors. The common ancestors proliferate into the different to the descendants with some mutations. The one proliferate descendant had no mutation, but others has. So then second generation and third generation and so on. Then finally we can check observe the situation. From the DNA sequence or some kind of the similarities can tell us the history about how they diverse during the history. This is a phylogenetic analysis. When we collect the samples and check the genetic diversity, and we can estimate the common ancestors, recent ancestors as like here, and the recent ancestors also have their ancestors, and finally we can reach out to the common ancestors of the total situation. This is a reconstruction of history based on the genetic diversity. Such a method is a molecular evolution. So if we have some samples, many samples, we can collect human and many kind of the species and many kind of interspecies. Even the interspecies, there are many genetic diversity. If we can get the genetic difference among them, that we can reconstruct the history. We can estimate the history based on the sequence. This is the final phylogenetic analysis. Probably many, there is, when the ancestors separated into the several descendants, unfortunately, some species has a weaker phenotype. in many cases, most of the cases, the mutation can cause the lack of function of genes. In such case, the descendant cannot survive within the ecosystem, so they disappear. But in some cases, they just meet difference. They cause just a difference, or some species, some subspecies may have stronger phenotype occasionally. Now, in such case, they can survive within history. But when the environment change, some species may become weaker, unfortunately. Finally, they also disappear. Another descendant can survive in some case. Then they can proliferate as like here. Then finally, the genetic diversity and the very rich genetic the life diversity can emerge. So that's entire history background of the diversity of lives and so on. Then anyway, the DNA mutation can cause the genetic diversity, and after the interaction among them, some subspecies can survive, some subspecies can have a mutualism, some subspecies can conflict. Such complex situation occurs and finally become the current situation. Then anyway, it's very important to estimate DNA sequence diversity, genetic diversity can include the information of the history and the situation of the why we can survive, why we can hear, and how they spread, and so on. That's like this is our example about the primates. The human is here, then the mutation, the speed of mutation rate is very stable in some species and so on. Then we can estimate the time the diverse. For example, the human and chimpanzee can were considered to diverse before 6 million years ago. Well, this means a million years ago. Then the common ancestors of primates diverse 63 million years ago, and some very primitive species still survive now. And this very ancestral species and human probably diverse the 63 million years ago. After that, some group, the New World monkeys, the diverse 40 million years ago, and we are a member of Old World monkeys. After about 205 million years ago, they diverse the macaca and some other kind of the species, and finally we become human. We now and then now the 2020. Yes. Anyway, during the history, the mutation accumulates within each species, and so such a mutation can tell us the background of what occurred within the history in our life. Within human, the humans, the Homo sapiens, and appeared within the horn of the Africa around here. After that, our ancestors migrate to the Europe and the Eurasian continent, and 70 years, 70,000 years ago, that we settled into the Asian regions, and then they moved to the American continent. the such history also can be estimated by the genetic diversity within our human species. When we analyze the human populations in each geographical background, such history can be reconstructed. Interestingly, based on such history, many genes changed. After that, some group of human ethnicity have some kind of advantage against the difficult environments, as like for example, the very northern area, the Eskimo regional groups have their special enzyme to digest the high fat foods, as like the whale and so on. Because it's very important to survive around their environments. And then the very highlanders, like Himalaya and other kind of some area had a high altitude. In such case, the pressure of oxygen is low, so that we need to carry oxygen more efficiently than lowlanders. So that some mutation accumulates related to such oxygen transmission in the blood changes, and finally they accumulate some kind of the special phenotype and so on. Actually, during the history, we have diversity. In some cases, some genes affect changes according to their survival history and so on. Anyway, such genetic diversity is very important to estimate not only our history, but also the environments where our ancestors survived in their regions. And more interestingly, some pathogens attached to us in history. This is a geographic background of mycobacterial tuberculosis, the pathogen of tuberculosis. As I told about that, they emerged in Africa as same as human. After that, the pathogen attached to the demography of humans. So that when we analyze the genetic diversity of pathogen, the situation is quite similar to human demography. Such relationship is also be found in another pathogens, Helicobacter pylori. This is one pathogen to infect with our intestine and our stomach. They also emerged within Africa as same as us, our ancestor. After that, they spread according to our demography. So this is the information of genetic sequence of Helicobacter pylori, not us, not human, but similarity can be found. So genetic diversity is a message of the history. And then when we collect many samples from our surrounding areas, that we can consider what they experienced and so on. So genetic diversity is very interesting as a research target. So anyway, this is the last topic about the genetic diversity. We can analyze genetic diversity based on the such difference because they have their own sequence, and such sequence can be analyzed based on the phylogenetic tree. For example, this is a simple figure of the genotyping. We have difference, as I told. Each difference can cause some kind of the difference. For example, there are many differences with us, so that it's very difficult to show the good example. For example, なんだ、そうね、sensitivity to some kind of the infectious disease. That in some group they have their own. They easily infect with some infectious disease, but others has a resistance. Such grouping can be done based on their types. If their changes are related to the background of genes, the difference can be correlated with the genetic changes. This slide shows such a concept. For example, phenotype A show within the three groups of genetic groups that the reason why the phenotype the reason of phenotype A may be based on the group 1 and group 2. Now, for example, phenotype C can be found within only group 2. That we can find some genetic background of specific difference within the variant group 2. So if the such classification may be, is correlated with the genotyping, genogroups, the such phenotype may be related to the genetic background. So this is more typical. That if we can estimate some difference. For example, some people is very good at painting. Such a phenotype, we can find many areas. And then maybe it's related to not genetic background. In Japan, there are many people who are good at writing or who are good at painting. Also in Indonesia, there are some people like writing, painting. In Africa, in Europe, in America, there are many such common types. But probably such group maybe is not related to the genetic background. In such case, we can find such groups, such types in many groups. Based on such grouping information, we can estimate the writing good at painting is not related to genetics background. But in some cases, there are some very tall height person may have to be connected to the genetic background. In such case, we can focus on what kind of genes may be related to the such phenotype. So that genotyping is very easy to estimate what kind of difference may be related to genetic background and so on. Then that's a genetic diversity, genotyping method in human genetics. Of course, anyway, the genetic characterization, genetic classification is very easy to estimate what kind of strains, what kind of personalities are similar or different one by one. This is a target when application of the public health. When we collect the pathogens one by one, in some pathogens have the same genotype. Others no. When they infect each other, the genotype become same. But if they are not connected, they are not infected with each other, the genotype will be different. Such information, such analysis is very useful to estimate the transmission routes of pathogens. So this research field is named the molecular epidemiology. So this is a concept of the molecular epidemiology. For example, there are 4 persons and infected with the same infectious disease within a school or within an office and so on. Then in such case, we can collect each samples, each strains from each patient. And then after that, we can analyze the genotyping one by one. Then if the 3 persons show the same genetic genotyping of pathogen, that we can estimate some outbreak cases, and then we try some kind of the control measures about that. But in other words, in other person have other case, other genotype, he or she may be infected from another area. So such information can be accumulated for the control of infectious diseases. We can also collect the many patients within some cities, areas, and so on. If they have some same genotype, they may infect with each other, but others not, and so on. Then such information can be used for estimation what occur within the pandemic or some outbreak within city, or in some case the hospitals or families and so on. This is one of the typical analysis of the pathogen. In some case, they have genomic DNA. It's very difficult to analyze the total genomic sequence because it's very huge. Then we use PCR. PCR is a technique to amplify a part of DNA. After the amplification of part of DNA, we can check the sequence one by one. If the sequence there are difference within the sequence, we can construct the phylogenetic trees or classify them into based on the gene.
Excuse me, sir.
Yeah.
You still have 2 minutes more.
2 minutes?
Yes.
Ready?
Yes, sorry.
Okay. Then this is the analysis of the SARS-CoV-2. It has already analyzed the sequence and COVID-19. There are mainly 6 parts and some part of the backgrounds. They are very. This is a current situation of the COVID-19. Based on the analysis, what we can estimate what occur around the world. So this is this become homework. Originally I want to show how to analyze them and how to observe the situation of the spread of the COVID-19 based on the genomic sequence. Now you can connect this here, and then finally you can find that as like the global situation. Then you can show the animation of the spread based on the genomic sequence and so on. Yeah. So this is the final slide. It's okay. The take-home message. So I want to share the interesting background of genetic diversity. Genetic diversity can be found not only between species but also within species. Such information can tell us the history of the species, and then we can use the analysis to control the infectious disease and some kind of species in the natural environments and so on. Anyway, thank you very much for the my opportunity to have lecture. Now, if any questions, don't hesitate to contact me directly. Sorry that we cannot check the chat in detail under the very short time. So that thank you very much.
Okay, thank you, Mr. Takayuki.
Yes.
Wada PhD from Osaka University. Arigatou gozaimasu. We are sorry for stopping you. And now, before going to the presentations of the last speaker, ladies and gentlemen, let's take a rest for a moment. And we are going to listen the beautiful voice from our director again. But now, she would like to entertain us with a song from Indonesia. Please give applause to the directors.
Thank you.
Thank you, Ibu Direktur. Give big applause for Ibu Direktur. Because she has entertained us with the song from Indonesia, so that we are not sleepy again. And okay, for all the participants, while listening to the next speaker's presentations, we will take the question from the chat room. So if you have a question, please write on the chat room, and don't forget to write your name and study programs and to whom the question is addressed. Ladies and gentlemen, now we invite our next speaker. Last but not least, Bapak Dr. Indrawan Permana Kamis, who will deliver a paper under the title Land Use in Central Kalimantan, an overview to planological concepts, engineer implementations, and impacts to natural environments. Time is yours.
Hello, everyone. Good morning. Thank you very much for moderator for giving this time to present. First of all, I would like to express my gratitude to Rector Palangkaraya University and also the Director of Graduate School of Palangkaraya University, who has given me a platform to present today, and also to our distinguished guest lecturer from Nagasaki University and also from Osaka University, Professor Hattori and Professor Wada. Thank you very much, sir, for your sharing today. We are looking forward for the next collaboration, mutual collaboration between Palangkaraya University and Nagasaki University as well as Osaka University. My session will be the last. I would like to present about land use. Professor Hattori say that about ecology, and Professor Wada explain about genetic diversity. Ecology and genetic diversity exists in our planet of Earth. Land use is an intervention, human intervention to planet of Earth. So it can influence ecology and as well as the diversity. My presentation today on the title Land Use in Central Kalimantan and overview to planological concept, engineering implementation, and impacts to natural environment. This is a subject in our doctorate program in Graduate School of Palangkaraya University in subject regional planning and environment. So I would like to greet to my students who also take part in this presentation. Okay, all right. I will start with this is the Central Kalimantan. As you may know that the size is 153,000 kilometers square. And population is only 2.2 million people, so is a low density. It means only 14 people in kilometer square. And if you see in the left side of my presentation, you can see the island of Borneo. And also called as a Kalimantan Island, the third largest island in the world. There are 3 countries exist in this island. One is Indonesia, another one is Malaysia, and another one also Brunei Darussalam. Land use is a product of planning. So planning is a kind of action of doing and how to do with a set of goals. So one of product of the planning is land use. Some I'm little bit explain just a little bit, not very in deep about the planning in theory. So planning is deciding in advance, not in the past, now, but in advance. What to do, how to do it, when to do it, and who to do it. So it involves also anticipating for the future and concisely. Planning is also goal-oriented. There are sets of goals that must be achieved by the action of planning. Planning is a primary function. Planning is all-purposive. It means that planning can be done in detail. For example, I would like to give you an example. If you want to move or to go from place A to place B, so you start planning, making a plan. And how to do that? You may think that you will use car. Okay, you will use car to move from place A to place B. Then you must think how to fill your car. You must go to the gasoline pump to get the gasoline. And also you must think how to get food. Yeah, because if you drive for a long time, you need to get rest and eating. So it means planning is all purposive. It can be done in very detail. Planning is also a continuous process. It's not a one-way process, but can be two ways, three ways, and can be done in a very long way. Planning also is forward-looking. It's always looking forward. We are using the past experience, yeah, in order to enrich our planning to achieve a set of goals for our better life in the future. Planning also involves choice. Yeah, there are a lot of choice alternatives that you have to choose, yeah, based on the best opportunities that you have. And also planning is directed toward efficiency. We must think about efficiency and effectiveness. And the product of the planning is land use. One of the product of planning is land use. So in this part, we learn that the land use, it can be defined as a land use as existed. It means in the past, it's already planned, but there is a spoiler. There is a misuse of the land use. And the plan is become useless. This is the one cases. Another case is a planned land use. You can go with the planned land use. Very good. Very, very useful for the future. And it needs to be done in the time ahead. So in this case, we define that land use can be defined as a land use existed and also planned land use. So it is a function or function that humans apply to the land available to them. So the study of land use is a study how the land is managed, how the land is managed, including how the natural world is adapted to human needs. So there is a needs of human, and also there is a land. So human think how to use the land to achieve their needs. Also, zoning is related to land use, but it is how government regulate and control it. So there is a government part, yeah, to regulate and to control land use. That's why when we creating land use, it is a multidiscipline. Yeah. We need to think about how to regulate the land use, how to make land use can be work, yeah, on legal basis. So that's why we need government to regulate and to control. It's a part of government. It's a policy. And this is the importance of land use. Yeah, it tells us about the great deal about the government making decision for land use and priorities they hold. And the better we can predict the future of the land use, the more we can prepare for negative context. So there is a type of land use. Yeah, is this for residential use, for settlement? It also can be used for transportation, for roads, bridge, so on, and institutional, open space, commercial use, and as well as agricultural use. Okay, I would like to show you the land Central Kalimantan province. We were talking about the strategic area. We are talking about the policy that given by government, three national strategic areas in Central Kalimantan, namely Bukit Raya Baka, Sebangau, and Tanjung Puting, and must be 13 provincial strategic areas. So you can see there is a 13 provincial strategic areas. It involves also the provincial interests inside. So this is a planned land use. It is done by law. Okay, if we see if we analyze the spatial pattern of the planned land use, so I would like you to see the purple color, part of purple color on the map. Particularly, I will show you here. This is a land that used for present land for preservation and conservation areas. The size is 5,400 21 kilometers square. It is called in local language as Sebangau. And on that area, there is height in natural diversity. So just to mention some of them, there are 505 species of flora and 35 species of mammals, 182 species of birds, and as well as 554 species of snakes, and etc. It also saved blackwater ecosystem. It saved peatland. And the most interesting is that some part of that area, which is called Sebangau, is managed by Palangkaraya University. We use it as a field laboratory in peatland. The area with the high natural diversity. Just show the potential of Sebangau. Yeah, you can see there is I have also already mentioned about the black water ecosystem. So there is some species like bird, mammals, as well as also forests. So the spatial pattern we concluded from the perspective of planological concept is used as a preservation and conservation areas, or we call it as a protected forest. By law, it is protected. And second, it is used for the cultivation areas that we can divide it as residential areas. It is used for the settlement. It is used for agriculture areas. So this is basically from the planological concept. Land use of Central Kalimantan is used for preservation and conservation areas, and also used for cultivation areas. And this is the spatial structure of the planned land use. As you can see there, that spatial structure of the land use from the planological concept consists of one is National Interest Activity Center. It consists of national interest inside. I mean national government. Second, it consists provincial interest activity center. And the third is local interest activities. So basically there are three kind of activity center inside of the land use of Central Kalimantan province from the planological concept. So planological concept is like this. Land use that was planned before should have a planological concept. So land use of Central Kalimantan, it is planned. So it has a planological concept. It was analyzed from different aspects using various tools of analysis, ranging from economic analysis, engineering analysis, social analysis, etc., and so on. It also contains a perspective of multidiscipline, ranging from economy, ecology, social culture, etc. It has set goals. It is clear for the land use of Central Kalimantan province. This is engineer implementation. Land use that was planned before should be implemented through set of policies. So policies is very important to direct the implementation of land use or a planned land use. In this case is central land use in Central Kalimantan. Next, in the midst of various topographic conditions, implementation of land use for residential functions should be engineered to find the best solution. So if the land use is aimed for residential function, so there must be some treatment or engineer treatment to make the land become ready for residential function. It should be engineered to find the best solution. For instance, land that often traps surface water should be engineered by making channels to drain the water, hence become dry and ready for residential purposes. Next, implementation of land use will influence natural environment. I have already mentioned in the beginning that implementation of land use is an intervention of human to natural environment. It will influence ecology and it influence biodiversity. There will be loss, gain. Impacts will be positive and negative because basically implementation of land use is an intervention to natural environment. That's easy to be understood. Impacts can be predicted or out of prediction. From the beginning, we predict like this, but after 20 years, the implementation of land use is going somewhere. It is out of prediction. At certain point, there must be some restoring action to recover the natural environment. Because when human intervene the natural environment, it will harm natural environment, then there must be such actions to restore or to recover natural environment for the next future. So this is a picture that in order to change the land to be residential function. Kalimantan Province, House Swam is engineered to be residential area. And also this is a forest. To drain the water out from the areas. Hence the palm tree can be grow on that area. This is an example of engineer implementation of land use, implementation of land use in Central Kalimantan. Like this. In this case, there is a cut and fill. They cut the hill in order to fill the valley. So there will be flat areas for residential purpose, or plantation purpose, or for agriculture purpose. So it will certainly make us suffer a huge loss of our valuable forests. This is a fact. And also for many times, smoke produced by forest fire covering Central Kalimantan harming picture. It is show that how the Central Kalimantan province, the whole of Provincial Kalimantan province, was covered by smoke. I think this picture was taken in 2015, year 2015. So it's covered absolutely the whole of the regional long time. And there is also devastating floods because of the forest is become lost, particularly in upper river. So it make water, directly drain to river and make river become full. So there is a flood surrounding the river, inundating villages as well as human settlements. Very much for your attention, moderator.
Okay, thank you, Bapak Dr. Indrawan Permana Kamis, for your explanations about land use in Central Kalimantan. Ladies and gentlemen, now we come to question and answer sessions. We already have the question collected from the chat rooms, but don't forget to fill out the attendance form absentee that we have shared in the chat rooms. Okay, in this session we give the opportunity to all the speakers to respond to the question sequentially. Mana pertanyaannya? But for Professor Mitsuru Hattori already answers all of the question in the chat rooms. And arigatou gozaimasu, Sensei.
Yeah, you're welcome, and thank you for raising my picture. Thank you.
Okay. Okay. Maybe you have another response to add your explanation here, Prof. Mitsuru Hattori. Okay. If there is not, so we go to the second chance for the Professor Takayuki Wada PhD and the chance for Dr. Indrawan Permana Kamis. Maybe you both have additional explanations for the questions. There are some questions that sent by the participants. We can decide the origins. Can we use gene mapping methods to do a measurements of genetic biodiversity? Something like this, the questions. If Prof. Takayuki Wada and Dr. Indrawan Permana have some additional explanations, please.
Sorry, it's my tongue. Yes. I
actually don't know which question that I should answer because I don't know the question.
Sorry. Okay, now we the host will share the some questions. some questions from the participants that we have collected from the chat rooms. But it is maybe about genotyping. The question maybe for Professor Takayuki.
Yes, yes. It's similar, but slightly different because the genotyping is based on the directly the mutations, and so we can estimate the different the targets not only for the population but also the each individual. Pedigree analysis is related to some kind of the population comparison, and anyway, the concept is almost the same. But the genotyping based on the sequencing analysis of some kind of the technique for the polymorphisms of the direct target of each genome. And the answer is partly yes. But the method is slightly different. All right. Is that okay? Can you listen? Hear my voice? Okay. Okay.
Yeah, I can hear. Yeah, but the organizer cannot. Organizer.
Recently, the sequencing techniques become more developed. So the situation during recent 10 years is quite different from previously. And I talked about the genomic sequence is difficult to monitor the entire sequence of bacteria, but it's not true. Actually, recently, the genomic sequence is very easily to be determined by the whole genome sequencing and next generation sequencers. Then the genotyping or some kind of the phylogenetic analysis become much more important for the biological research.
Okay, this is from Prima Jaya. There is I have question for Takayuki. What is your question, Prima Jaya? Is there any question from Prima Jaya?
Which question?
Okay, while waiting, there is another question from Berkatni Agus Triana Intansari. Economic and natures always grow in contrast. How do we deal with it? What is your opinion about how the world must see the forest as the value of money, of the resources of our life? This question for any speaker who is willing. Maybe Dr. Indrawan Permana would like to answer it.
Yeah, thank you very much, Moderator. So I think this question goes for me that from Berkatni, economic and nature always grow in contrast. Yes, it is. Because when we talk about economy, we talk how we can exploit the natural environment and change the natural environments, commodities of the natural environments to be money, and money can be used for development. But in the other side, nature will suffer from the exploitation. So if you think about the economic and nature always grow in contrast, yes, but I would like to say that this is the old fashion of thinking. Because in the past, yes, economic and nature will be always grow in contrast. But now we cannot think like that, because economic and nature should always together. It should always together. So that's why we should think about the sustainable development. So we can deal with nature. In one side, we can get development. We use the natural environment, but in the other side, yeah, we can also preserve and conserve nature. We can go together as a counterpart, economic and nature. So in the future, we can see that our forests can be conserved, can be served to the human, to the society. That is my my response. Thank you very much.
Okay, thank you, Dr. Indrawan Permana. There is questions from from Jaya. If I don't make mistakes. Wait a minute. Mana tadi lah? Ada tadi. Simit. Ada Jaya tadi mana bertanya? I believe is there any specific analysis for genotyping? Maybe it goes the question is goes for the Professor Takayuki Wada, PhD, maybe. Prof. Takayuki, could you please to give a response for the question from Prima Jaya? Is there
I
any
cannot
specific?
identify which question should be answered. Can you show the?
Tuliskan pertanyaannya di sana. Ini nih, saudara. Wait a minute, sir. We will share the question for you. Could you see it?
Yes.
Okay.
Specific analysis. Specific. What means a specific analysis? There are many kinds of the specific analysis. This means the method for the species separation, or I cannot understand what the conclusion means. That then if we want genotyping mainly used for the with analysis within species, then when we want to make the phylogenetic tree between the species and the species, we always use the genetic sequence. In such case, we don't say the analysis as genotyping. The research, the analysis is named the phylogenetic analysis or molecular evolution. So the genotyping may only focus on the very small difference within the species when we want to find the relationship within the very recent history among the species, as like the transmission or some kind of the correlation between the disease and some group of human individuals and so on. Then it can be considered as answer. Sorry.
Okay, thank you, Mr. Takayuki, for the response. But it is still there is a question from the Disaputra. Good afternoon, sir. About two female scientists received the 2020 Nobel Prize in Chemistry for discovering technology for editing DNA, known as the genetic scissors, CRISPR, Cas9. What do you say about making specific and precise changes to the DNA residing in living cells? We would like to share the question. Okay,
Yeah.
that is the questions.
This is just for the technique to modify or change the genomic sequence directly based on our own purposes. This is not the kind of the natural situation. So that we cannot, the genotyping always used for understanding the natural situation of the species. And so that, this some kind of the gene targeting and CRISPR-Cas9 directly change or modify based on our hypothesis. So the direction is opposite. Okay. Nah, genotyping
Ada
or
pertanyaan
some kind of phylogenetic analysis
lain.
is to understand the natural situation. But some genome modeling or some knockout mutations are used for the verification of our hypothesis. So that's named as reverse genetics. Reverse genetics. When we analyze the natural situation of genotyping and the genetic situations, that's named as genetic. So the direction is opposite. The knowledge or the techniques we should learn is quite similar, but the direction is quite different. It may be related to the central dogma that it's the direction is DNA to the protein. But if we know the function of protein, we can reverse to the DNA. That means why the reverse or direction is opposite. Okay. Is that okay as an answer about this?
Yes, all of the questioners has satisfied with your answer, sir. Okay, ladies and gentlemen, after listening to the whole of the lecturers, now we are more realize how is the importance of ecology preservations and other elements which support it, like genetic diversity and land use. Ladies and gentlemen, our international public lecture today is very interesting, but because we were limited, we finally reached the end of the events. Before we close, we have to take a picture first. We will take from slide by slide. Okay, please host, take a pictures from slide 1 first, and keep smile and cheers. Next slide. Keep smile and cheers. Okay, finish. Thank you. Ladies and gentlemen, now we come to the end of this event. I would like to thank all of the honorable speakers who share their knowledge on this opportunity, and also thank to all participants, especially students of postgraduate programs of Palangkaraya University and Nagasaki University who had participated actively. It has been our pleasure to host this event, and we hope you all have a pleasant day. See you on the other event, but before we leave, our director will sing a song again for us, entertain us for the last sessions. What song is it, Ibu? Kokoro no Tomo. Okay. The director will sing for especially for Professor Takayuki Wada and Hattori. Okay, sing together. Wait. Thank you, all of the participants and honorable speakers. See you on the other event. Thank you. Arigato gozaimasu.
Arigato gozaimasu. Thank you very much for Hattori and Professor Wada. Thank you very much, sir.
Thank you, everyone. Nice to meet. Have a nice day.
Have a nice day.
Bye-bye.
Bye.
Gila, bagusnya sukses. Itu sudah rekaman tadi lah. Matiin kah? Boleh.